Okay, so we just went over the kind of the normal pattern of DNA methylation, normal function and pattern of DNA methylation. Let's look at how DNA methylation behave in diseases. We use cancer as an example. Okay, so cancer is a genetic and an epigenetic disease, right? So we all know it's the genetic part, but it's also an epigenetic disease. And there are a ton of crosstalk between these two types of mechanisms, right? So for example, um, the two most important hallmark of cancer in the context of DNA methylation is this genome-wide hypomethylation and increased focal methylation over promoters of tumor suppressor genes, right? So as we just mentioned that when you have genome-wide hypomethylation, the paracentral macroregions are losing DNA methylation, right? And they're filled with repeats, right? And these repeats are actually perfect subjects for recombination, right? So this might lead to recombination to happen. You're gonna see deletions, amplification, copy number changes, right? So the genome becomes fragile. So this is sort of an epigenetic change leads to a genetic change. Increased methylation over tumor suppressor genes, for example, DNA repair genes, right? Will shut down DNA repair genes, and then cancer cells start to accumulate mutations, right? So this is also a event where epigenetic changes can lead to genetic changes. And vice versa, we also see a lot of genetic changes, mutations in enzymes responsible for chromatin biology get mutated in cancer, right? So by and large, tumor genesis is a process of interaction between genetics and epigenetics. We know this, for a long time. In fact, a lot of the knowledge about DNA methylation was gained from studying cancer. We know this two-hit hypothesis, right? So this is actually how a lot of tumor suppressor genes are identified. Turns out DNA methylation is a major, probably the major mechanism for shutting down tumor suppressor genes, right? We know, genes have two copies, right? So in cancer, you need to lose both copies of tumor suppressor genes, right? So sometimes, cancer start with the first copy being lost through a deletion, and then the second copy can be lost by mutation or another deletion. So this is where loss of heterozygosity is defined, or by a DNA methylation, right? So these are the mechanisms for taking out tumor suppressor genes. So a lot of tumor suppressor genes were actually discovered by identifying this abnormal methylation pattern. So this is a recent example where people show, how epigenetic event alone can lead to tumor genesis, right? So this relates to how chromatin is wrapped around in the nuclei. I believe you will have a lecture on that, right? So here, the chromatins form topologically associated domains separated by domain boundaries, usually bound by CTCF, which is actually a mesosensitive protein. So here, in normal situations, CTCF binding sites are unmethylated, so CTCF can bind here. These two domains are separated, they're doing their own business, they don't talk to each other, right? But in cancer, sometimes the CTCF methylation level goes up, and then CTCF doesn't bind anymore, right? As a result, these two otherwise unrelated domains start to interact. But in the domain on the left, there's a strong enhancer, and in the domain on the right, there's a oncogene, right? So now this enhancer touches this oncogene, resulting oncogene upregulation, and that actually leads to glioma, right? So this is a example where epigenetic event can lead to tumor genesis, and really combines oncogene, demethylation, chromatin domain, all these concepts together. But the typical pattern in cancer, we already actually talked a little bit, is now the tumor suppressor gene promoters are often methylated, right? So again, this is a very common, probably the most common mechanism for cancer to shut down tumor suppressor genes. The other one is this global demethylation, right? So the centromeric regions are losing demethylation. So these are the patterns of demethylation in typical cancers, right? So just to summarize, there's a very nice review out there, multiple reviews out there. Right? So in cancer, you see increased methylation over tumor suppressor genes and decreased methylation over paracentromeric heterochromatin. This leads to genome instability. This leads to loss of tumor suppressor gene. That leads to cancer, right? So this is the kind of pattern we see. This pattern actually gives us very important hint, right? So now let's look at these tumor suppressor genes. We know cancer needs to do two things: activate oncogenes, silence tumor suppressor genes. You can silence tumor suppressor gene by mutating it away, deleting it, right? But you can also just methylate its promoter. If you mutated the tumor suppressor gene or deleted the tumor suppressor gene, there's nothing you can do, right? But if the tumor suppressor gene is methylated, the gene is actually still wild type, right? So this actually gives a hope. Maybe this is a new way to treat cancer cells. Well, it's not new anymore. So this is actually a fairly hot field for about 20 years now. People have developed this epigenetic cancer therapy using, for example, DNA methyltransferase inhibitor. You can reduce DNA methylation. Hopefully that also happens on tumor suppressor genes and reactivate tumor suppressor genes, right? So this has been in the clinic for many, many years, 20 plus years. It works somewhat, right? So it works a little bit for blood cancers. In fact, for MDS, this is FDA approved therapy, but it doesn't work for solid tumor. I mean, it's on and off, right? So, the field has gone from, being very, very active to a little bit unsure, but just the past few years, the field sort of is revived for very interesting reason and also related to, transposable element. Right? Turns out, activating tumor suppressor gene is not the only, maybe not even the major mechanism for the therapeutic effect, right? So this type of therapy is not specific. It doesn't target tumor suppressor genes. So it actually acts on the entire genome, right? So as a result, it's not just the tumor suppressor genes potentially getting expressed, the genome getting, the whole genome is getting transcribed. We kind of already said what's the most abundant stuff in the genome. Transposons, repeats, right? So let's say you have repetitive sequences, in antisense orientation, right? Transcription goes through and you ended up getting RNAs that can hybridize and form double-stranded RNA, right? So when the cells sees double-stranded RNA, the cell will think, I got a viral infection. They're going to activate immune response. So people now believe that epigenetic therapy actually primes cancer cells for immune response, right? Now, for patients where the immune system is weak or the cancer has evaded the immune system, epigenetic therapy doesn't work. But for cancers, for patients where you have stronger immune system, maybe it will work, right? So this gives people a hint that maybe we can now combine epigenetic therapy with immunotherapy to improve cancer therapy, right? So there's multiple clinical trials going on to test this hypothesis, okay? I'll just wrap up this part by just telling you, demethylation is not just implicated in cancer, it's everywhere. It's implicated in many other diseases. I'm gonna quickly go over these two related but very different diseases that are, we call imprinting diseases. So Angelman syndrome and Prader-Willi syndrome, two very devastating diseases. They're kind of related, but they're, phenotypically, they're very different. These are genetic disease, so people have mapped where the problem is. The interesting thing is these two diseases got mapped to the exactly same genetic event. They got mapped to the same deletion on chromosome 15. So that's interesting, how the same deletion result in two very different diseases. Turns out, within this 4 megabase region on chromosome 15, there are two imprinted genes. right? One is maternally imprinted and the other is paternally imprinted, right? So depending the deletion happening on the paternal chromosome versus maternal chromosome, the effect is you're losing different genes, right? Imprinting is interesting, but imprinting also tells you that, we lose this redundancy, right? So one copy is already silenced. It takes just one more hit to render this gene useless, right? So this is why imprinted regions are enriched for human diseases. And this is also why the same deletion, right, have very different effect happening depending on which chromosome it happens on, right? So this is just another anecdote relating DNA methylation with other diseases. Okay, so this is a uh, good place to stop recording.